Hello students. Today we will discuss about the fermentation and fermentation system. So we will start from the fermentation, that what is the fermentation? Fermentation is a metabolic process in which sugars are converted into the acids, gases and alcohol. Suppose you have a sugar that is glucose. Assume glucose is your sugar, then you know the glucose is converted into the pyruvic acid by the process of glycolysis, which is a series of biochemical reactions, and this glycolysis takes place in cytosol. So glycolysis occur either oxygen is present or oxygen is absent, so it is completely independent of the presence or absence of the oxygen. So here glucose or sugar is converted into the pyruvic acid. Further, there are two fates to go for this pyruvic acid on the basis of absence or presence of the oxygen. When oxygen is present, then this pyruvic acid got converted into acetyl CoA, acetyl coenzyme, and which finally enters into the Krebs cycle, and which is the part of the aerobic respiration. And when this oxygen is absent, then this pyruvic acid is got go under to two processes. First one is fermentation and second one is the anaerobic respiration. So what is the difference between these two, fermentation and anaerobic respiration? Just in simple, in case of both takes place in the absence of oxygen, but in case of fermentation the final electron acceptor is an organic substance like your lactic acid, ethanol, and in case of this anaerobic respiration, the final electron acceptor is any inorganic substance like sulfates, nitrates. But oxygen is not here the final electron acceptor, because oxygen does not take part in this process. Oxygen take part in the aerobic respiration. After that, this fermentation process is further is of two types on the basis of final electron acceptor. Further, if lactic acid is the final electron acceptor, that then the pyruvate is converted into the lactate, and further pyruvate is converted into the ethanol under the ethanol fermentation. And in ethanol fermentation, ethanol and carbon dioxide as a byproduct are produced. And in case of lactic acid, this fermentation, lactobacilli, some fungi are responsible for their conversion. Further, this fermentation system is of three types, that are the batch fermentation, feed batch fermentation, and fed batch fermentation, and continuous fermentation. So before we study about these systems of the fermentation, it's very important to study about the growth curve of the microorganisms. Before this study, this growth pattern of the microorganism is very important, because these further systems totally depend upon the growth pattern of the microorganism. So microbes can be bacteria, fungi, viruses, but most of the time the microbes is generally in the industrial microbiology mostly are the bacteria. So we don't if we don't know their growth pattern, we can't proceed to the fermentation process. So here we'll study the growth curve of the microorganism, especially of the bacteria. You can see here this curve. We have plotted a curve. This one is the x-axis, this one is the y-axis, and here in this curve you can see that there is a plot like this, just like an this somewhat like a shape, matlab like this, some but is stationary, some but is where again there is a exponential, again after expansion there is a straight line, and after that again there is a fall down. So in this growth curve, toward the x-axis there is the time, and toward the y-axis there are the number of bacteria, or the biomass of the bacteria. So this one here you can see that these red color dots up to this point, this whole is the log phase. Log means it is just like in halting phase. Halting means when something come and stay for some time, and means it can, we can also say that it is a preparatory phase. It is that phase in which further growth do not take place. They ceases up to some extent. Don't, we can't say that ceases, but they are in that state that they are taking rest. They are judging the environment. They are judging that environment, that if further environment is favorable for the growth of the microorganism, they will flourish, they will grow. But if environment is not suitable, they can be in the long phase. For example, when you culture any bacteria and you transfer that bacteria into the new battery plate, there is a media in the battery plate and you transfer in a new media, so bacteria takes some time, 5-6 hours. There is a time in between, there is no division of the bacteria, and after that immediately bacteria appear, and that halting time, that time is known as the log phase. After that, this phase is also known as acclimatization phase, and in this phase bacteria are no much eager to grow. Further that, there is a lag phase. Lag phase means, you can see that this is also known as the exponential phase. Why exponential phase? Because bacteria grow very fastly in this phase. There is a complete growth of the bacteria in this phase. They grow, means the number of cells also grow with the time. As the time increases, the cells also increases in the number, cell also increases in the size. And here we measure the amount of the cells by taking their optical density, and after that through spectrophotometer we observe the observation and we plot the graph further, and that graph gives us the straight line, means bacteria are growing very fastly, like this. Further that, we'll obtain a straight parallel line, and we can see that this phase is known as stationary phase, because here the bacteria which are growing very fastly, they come to an somewhat complete leveled phase. Leveled phase means in this phase there are limited space requirement, there are the limited supply of the food nutrients, so the level became in constant, in which is known as the stationary phase. After stationary phase, this one is the post stationary phase. When the bacteria enter into this phase, the you know, the amount of bacteria is continuously increasing here, and the stationary phase amount we come up to that level. And after that, in same time and same situation, food is limited, nutrients are limited, space is limited, so what will happen? Some of the bacterial cells start dying because of the scarcity of the food, scarcity of the that type of the space. Some of the bacterial cells produce toxic substances. Those toxic substances kill the bacterial cells themselves. So due to this reason, the cells decrease in the number, and this one is the post-stationary phase. And finally, there is the death phase. But in case of microbiology, death phase we never count death phase, because it is not of any economic important. We generally talk about the lag phase and stationary phase. These two phases are very, very much important from industrial point of the view, from production point of the view. So if we talk about in case of microbiology, we always want to reduce this lag phase, this preparatory phase, because we want maximum product in less time and in less cost. So there is always a need to low this log phase. So further, this lag phase is very important, because in the this lag phase, various alcohols, various beverages, just as alcohols and other